Hi, welcome to another lesson with Dr. Hill. Today we're talking about biology and genetics, and we're going to talk about the predictions that we make from Punnett squares and why those predictions don't necessarily come out in reality. Uh, for example, I might have a gene for tail length in mice. We've talked about this before, where big T gives me a long tail, that dominant allele, and little t, if it's present as a recessive, gives me a short-tailed mouse with a little stumpy tail. Now, if I do a particular cross, I might be able to predict that all of the offspring are going to be a certain thing. All right, so let's just have a quick look at that cross on the board here. If I look at that cross and we see here big T, little t, big T, little t, and I cross that, 25% of the offspring should be little t, right? Okay, so I get 100 offspring, 25 of them are going to be little t, right? They're going to be short-tailed. Well, you see, that's not necessarily true. For example, I might just have 10 offspring. Let's give these mice a bit of a break and just have 10 offspring. 9 of them are long-tailed, 1 of them are short-tailed. Does that mean I'm wrong here with my predictions? No, it does not. What it means is that genetics is random. And the way that the various alleles are segregated into sperm and into eggs is a random process. And it's entirely possible that 9 sperm carrying the dominant allele will fertilize the eggs, and only 1 sperm carrying the recessive allele will fertilize the eggs. And that's just random chance. Okay? So we need to think about more like rolling a dice. If I roll a dice, there's a 1 in 6 chance it's going to roll a 6, right? So if I roll the dice again, there's still a 1 in 6 chance I'm going to roll a 6. Now, I'd probably give myself a better chance of getting a 6 if I roll 6 dice, because each one of them has a 1 in 6 chance, and one of them's bound to roll a 6, right? And you could say that for any number. So let's just do it and see. I'll take my dice here. Let's see. I've got 6 dice. No, not cheating. 6 dice. Give them a good shake. And let's see. I should roll 1 of every number, right? Because there's a 1 in 6 chance of each number. Okay. Well, I rolled 2 6s, so that's unlikely. I rolled 2 5s. I rolled a 3 and a 1. I didn't roll any 2s, and I didn't roll any 4s. So how many times do I have to roll the dice to get a 2 or a 4? Let's see. That's rolled it 6 times already. And now we've got 2s, 4s, 1s, no 5, no 6. So what I'm trying to demonstrate here is that we can make predictions about the probability of an outcome, but reality is never necessarily going to match it. I mean, it's entirely possible I can roll 6 dice and I get 1 1, 1 2, 1 3, 1 4, 1 5, and 1 6. Absolutely. And over many, 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 many rolls of the dice, the number of times the 1 shows up, the number of times the 2 shows up, the number of times the 3 shows up are roughly going to be equal. They'll not be exactly equal, probably, because it's random. And we have to think about genetics as being a random process. There are other reasons why we get variations in the offspring. One of the reasons might be that a particular phenotype is lethal, and that after the fertilization that produces that phenotype, the embryo doesn't develop and just dies, in which case we'll never see that offspring. So there's a number of other possibilities for this. But what I wanted to do and just show you in a very quick short lesson is that the probability that we predict is only a probability. We cannot necessarily expect those numbers to be exact. In fact, some people look back at Mendel's work, and I'll link you to a video on Mendel, who's the kind of father of genetics, and did also experiment with pea plants. There's some people who've looked back at his results and thought, well, they look a bit fishy because they come out so neatly. But the key is, in order to be sure about these things, it's like rolling a dice. You need to do it over and over and over again, collect lots and lots and lots and lots of data, because it's a random event. And to be sure, if you really want to analyze this and show that it's a 25%, you need to do an awful lot of crosses to get that number as close, because the reality is that it's not going to come out that way you know, when you do it with just a small number. All right. So, I mean, think about families, you know, as the other possibility. I mean, there's a 50% chance of a girl and a 50% chance of a boy, right? 50-50. But you'll know families that have 3-4 sons and no daughters. And you'll know families that have 4 daughters and no sons. And you'll know families that have 2 daughters and 2 sons. But it's still a 50-50 chance. Every time somebody is born, they have a 50-50 chance of being a male or a female. And yet we have families where we're all sons or all daughters. Okay, so remember, reality doesn't match up with our probabilities, our predictions, because our predictions are based on an understanding of a model, of a system, but reality is random. All right, so we're trying to predict the outcome of a random event, and that's why the reality is never the same as the probability. Okay, I'm rambling now, so I'm going to stop there, and hopefully I'll see you in the next lesson.